Hi, I'm Manoj. First of all, I would like to thank the organizer for giving me this opportunity. Am I audible now? Okay. So, yeah, first of all, I would like to thanks to the organizers for giving me this opportunity to present my work. I'm Manoj. I'm working with Shashi at NCBS. I'm a postdoc. So today I'm going to talk about self-organization, self-organized dynamics of freely jointed active droplet chain. So our interest in active assembly is basically we are inspired by the biology to create active molecules where we can create a new dynamic materials. With active polymers, we are interested in studying the new physics by activity-driven non-equilibrium fluctuations and conformation changes. And broadly, we are motivated by the folding in protein, and we are attempting to create a freely jointed linear structures which can undergo activity-driven folding. So, for example, there are examples of these theoretical examples of these active filaments, and there are very rare experimental examples. For example, these active these are the active chains of colloids, and the problem with such kind of system is they have fixed internal orientations and they are not freely jointed, and they are but whereas we are going to develop a system where we can control the internal orientations as well as the bonds can freely rotate. So the kind of system we are using here is a simple oil in water system, and where these oil droplets are stabilized by the surfactant at a concentration below critical micellar concentration, but above critical micellar concentration, these droplet start self-propelling, undergoing micellar solubilization. So the self-propulsion is caused by the self-sustained gradient in surface energies, which generates Maragoni stresses and induces the flows. And so here we are using 5 Cb liquid crystal as a oil phase. So just to remember that the cores of these droplets are also liquid, and they have these pneumatic directors, but when they start propelling, okay, so the internal the convective flows generates inside these droplets, and these defects get aligned in the direction of self-propulsion. So to visualize this whole process, I'm going to show you a video. Okay, so here we use oil soluble dye, and initially it creates a cloud of filled micelles. Once it breaks the symmetry, it starts propelling and generates a trail of filled chemicals or filled micelles. And yeah, so these droplets have the self-sustained concentration gradient between filled and empty micelles, and yeah, so with the when they generate this trail of filled micelles, these such droplets avoid crossing not only their own trails but also the trails of neighboring droplet. So that shows their repulsive autochemotic nature. So such kind of such kind of microtumor are the physical realization of so-called Squirmer models, okay, in which the hydrodynamic flow fields are described, in which the hydrodynamic flow fields are described by the slip velocities. Okay, so here I'm going to show you that these slip velocities can be controlled by controlling the fraction of filled micelles. So without going much into detail, I'm going to show you here when we change this fraction of filled micelles in such microsimmer system, we see a qualitative differences in the flow fields. So here, this is a dipolar flow fields of these microsimmers, which becomes a quadrupolar as we increase the fraction of filled micelles, and which are extensile in nature at 20%, and which becomes contractile in nature at 70% fraction of filled micelle. And since there are chemical fields set up along with the hydrodynamic fields, so we also see qualitative differences in the chemical fields of these microsimmers. So since with the increase in the fraction of filled micelles, okay, we see a change in the slip velocities, but at the same time, we also see that the propulsion speed of these microsimmers also decreases. Okay, but we also see at the same time qualitative differences in the trajectories of these microsimmers, okay, which are partly self-crossing, and as we increase the fraction of filled micelles to 20%, they become self-avoiding, and further they start making propelling straight at the 70% fraction of filled micelles. So now I'm going to show you. So when we use the formation of uniform polymer chains by using such microsimmer system. So since we want to form these uniform chains, so basically we need to have a uniform size droplets. So we use a microfluidic technique to generate a uniform size droplet, so which can be seen here from this bright field microscopy image, that all the droplets are uniform in size. So here, instead of using only surfactant to stabilize these droplets, what we use is basically mixture of lipid and surfactant. So the reason why we want to use here lipid along with surfactant, because we have this biotin at the surface of lipids. So by using this biotin, we can create a biotin streptobin chemistry, and so that we can create a bond between adjacent droplets and form these such kind of linear assemblies. So the presence of lipids and the presence of streptobin on the surface of these droplets is confirmed by the fluorescence microscopy, where we basically use a fluorescently tagged lipids, and so to generate assemblies by using such droplet system, what we take a two fraction of droplets. The one fraction in which biotin at the surface of the droplet is saturated with the streptobin, and the other fraction of the droplet in which we have only biotin at the surface of these droplets. So when we mix these two fractions together, so we generate these kind of linear assemblies. So these random configuration of these linear assemblies already confirm their freely jointed nature, but to confirm it more, so what we do, we basically make these droplets active, these chains active. So they show their clearly freely jointed nature, and these chains are propelling along perpendicular to their body axis. Further, what we do, basically we plot angle theta between three adjacent droplets, and all possible angles for the theta are plotted in the scatter plot. We can, along with these chains, freely jointed chains, which are propelling perpendicular to their body axis, we can also create a chains which can, can I go back? Yeah, so we can also create such freely jointed chains which move along their body axis. So So here I'm not going to talk about this, but we are going to, here I will be mostly talking about these chains which propel perpendicular to their body axis. And so when we take these chains in a completely isolated environment, with isolated I mean where there are no any kind of droplet or assembly surrounding these droplets, these assembly, they actually have this C-shaped configuration, and their propulsion is basically ballistic in nature. So when we measure the flow fields of these linear chains, so they have this dipolar symmetry at their ends, but when we change the fraction of these field micelles, we see these vortices in front of these self-propelling chains. I just changed the fraction of field micelles. Yeah. So whereas the shape of these chains stays stable, and which we confirm by measuring the bond angles. And so if you remember that by changing the so we can control the slip velocity of a monomer just by controlling the fraction of field micelles. So here we see a reflection of change in the slip velocity in the flow fields of these linear assemblies. So here at 0%, we don't see these external flow, these extra flow vortices, whereas at 70%, we see these extra flow vortices. And since we know there are associated chemical field along with these flow fields of these linear structures, so these flow fields are basically symmetric in nature. At the same time, the chemical fields are also symmetric in nature. Whereas at 70%, we see a asymmetry not only in the flow fields, but also in the chemical field. So this also shows that these chemical fields and flow fields are shaping each other. So whereas in case of this 70%, we have so over a time, in case of 0%, these flow fields and chemical fields symmetry basically is time independent, whereas at 70% it is time dependent. So because of that, we observe basically oscillatory dynamics with the fraction with the increasing the fraction of these field micelles. So whereas this is time dependent behavior of this chemical field in case of these linear chains, what we see is basically there are oscillations which are generated due to the interaction of these linear assembly with this own chemical field. So as if you remember in the beginning, I told you that these droplets are basically repulsive, they have negative autotactic nature. Because of that, when they actually interact with their own chemical field, they generate these oscillation in their dynamics. Okay. Okay, so we actually create these freely jointed chains of these active droplets, which can propel both perpendicular to their body axis as well as along their body axis, and we can tune the slip velocity by controlling the concentration gradient of field micelles, and we show that hydrodynamic and chemical field shape each other, and we observe these oscillatory dynamics of these active droplets. And apart from that, so we are also able to generate structures which are closed and branched and which have these interesting dynamics. Okay, such as for dimer, they have like run and tumble kind of reaction. Here they act as like a pusher to these assembly when they self-propel. Thank you.
Manoj. Questions? Rajesh and then Roshan.
Yeah. Very mesmerizing videos. So I had just one question. Since you're using the streptavidin biotin chemistry,
yeah,
how is it that you always don't end up with branch structures and you also get linear structures?
We do get, but the thing is we can separate them out.
Oh, okay.
Control the length of chain actually.
We don't control the length of the chain. Basically, yeah. So maximum length we got is like 13 mer.
And if I have understood correct, you have studied at 0% FMC and 17%.
Yes.
What happens in between? Like, or
So we get, so if you remember, I have, so you're talking about in case of chain, or you are,
no, like, yeah, in case of chain only,
Yeah.
you have shown the flow field, right?
Yeah.
At 0% at 70%.
yeah,
So 70% is the limit or the critical value beyond which it happens,
Yeah.
or there is something in between the.
yeah, at 50% also we get almost similar behavior.
Okay.
Yeah.
Okay, Mahesh, last question in this talk.
So under what conditions do you get motion in the axial versus the transverse direction? How do you change? What is changing?
Okay, so here we change the surfactant concentration. Yeah.
Thanks, Manoj.